Right, we're finally on to the last of the GI secretions. I know you guys have a lot to listen to. There's a lot to say too. All right, so now we're on to the level of the pancreas. And as you saw, the pancreatic secretions are stimulated to be released when the intestinal phase begins. Two of the main products we discussed were secretin, which is stimulated by acid presence in the duodenum. When fats are in the duodenum, that causes the release of cholecystokinin, can, which will activate the gallbladder so that it contracts. And we'll look at the physiology for that. If you pretend this green box is the liver, the gallbladder is right there, storing bile, about 50 milliliters of bile that will contract. There is a pancreatic duct running through the pancreas, and the common bile duct from the liver gets together with the pancreatic duct. And the sphincter of Oddi is a sphincter that controls the release of products from the common bile duct. So when CCK causes gallbladder contraction, that will force the stored bile into the common bile duct, then into this shared duct here, and then that will cause the pressure to go up and hopefully the sphincter of Oddi to relax so that the products from the pancreas and the liver can enter the duodenum. Vagal stimulation causes release of pancreatic enzymes. Remember that vago-vagal loop. So there is mechanoreceptor innervation going up to central areas, which then in response will cause acetylcholine release to activate the cells of the pancreas. All right, so secretin causes the release of bicarbonate, so that is very important. Without bicarbonate, we don't get the acid entering the duodenum neutralized. CCK is going to drive the secretion of enzymes and gallbladder contraction. All right, so looking at the anatomy of the pancreas has a tail region and it has a head portion. The head portion is what comes into contact with these ducts and with the duodenum. Because it has free access to these places, that means that the head of the pancreas is most likely the one to become damaged. Pancreatic cancer almost always begins at the head of the pancreas because of its anatomical location. So the pancreas has a couple of different types of cells. In a later chapter, we'll go through its endocrine function, but the cells that are endocrine in nature, meaning they make products and dump them into the bloodstream, are located in the pancreatic islets. They're also called the islets of Langerhans, named after the scientist who discovered them. There are A cells and B cells, alpha, beta. Beta makes insulin, alpha makes glucagon. Insulin lowers blood sugar, glucagon increases blood sugar in fasting states. Then we have acinar cells, so these little clusters of cells. They are secretory cells, so they will secrete their products into this duct, which will converge together to make the pancreatic duct, which will then empty into the duodenum. Again, I want you to note that the common bile duct merges together with the pancreatic duct, and flow of both of those products, pathways, are controlled by the sphincter of Oddi. When that's open, we can have release. When it is closed, things remain in there. Again, the endocrine function does have a role to play in eating, but right now we're talking about secretion, which means that we have to have our exocrine products, so things that are emptied into a duct. The exocrine function is to make what we call the pancreatic juices, which we'll see on the next couple of slides. And the pancreatic juices converge together to make this large juice profile that's added with bile into the duodenum. This is what our acinar cells look like within the pancreas. So here's one single acinus. This is a duct cell, small duct because there's an accessory duct to the pancreas as well. Those secretory cells will spit out their products through exocytosis into this duct area so that it can be released. Here are your enzymes and juices. And this picture I like. It puts everything together with liver and pancreas. So the liver we know is making bile, and that will be stored in the gallbladder if there is one. When people have their gallbladder removed, they have to stick to a low-fat diet because you need a lot of bile to break down fat. Together with the bile come the pancreatic juices. There's three which you should know if you don't already. There's amylase, which you've seen a lot. Amylase breaks down starches. So pancreatic amylase breaks down starches into maltose. There's trypsin, that is your peptide one, so that digests proteins. And pancreatic lipase breaks down fats into fatty acids and glycerol, which can be utilized in a variety of bodily processes. All right, so your trypsins are the ones that we consider important, and they're also the reason why the pancreas can be damaged and why things like cystic fibrosis, pancreatic cancer, pancreatitis are so deadly because it digests proteins, and organs are proteins. Trypsin and chymotrypsin, they split whole proteins into smaller peptides, and then carboxypolypeptidases, little enzymes, they split the peptides into individual amino acids so that the liver can deaminate them. We already said amylase, that helps break down our starches, glycogen, all carbs except cellulose, which are the components of plant cell walls, so that will be incorporated a lot of the time into the roughage of the stool. Lipases, neutral fats into fatty acids and monoglycerides. There is cholesterol esterase, which hydrolyzes cholesterol, and phospholipase, PLA, breaks down fatty acids into the component phospholipid portion. All right. There is a small amount of trypsin inhibitor located essentially in the pancreas. However, if that is overwhelmed, generally by a blockage, so that could be either due to swelling, like in the case of pancreatic cancer, there would be a physical swelling here impeding flow. This can also happen if there is a gallstone, which becomes impacted in the common bile duct. In the case that trypsin inhibitor is overwhelmed because of a blockage or something not allowing the outlet of pancreatic juices, what happens is the trypsin becomes trapped within the pancreatic duct, where it then becomes activated and then starts digesting the pancreas, because that's its job, to break down proteins. So here is a picture of what pancreatitis looks like, which is inflammation of the pancreas. You can see when this is activated, it causes a huge amount of damage to the surrounding areas. So here you see pancreatic damage, leaked trypsin, and you see ischemia of the small, I can't even speak anymore, the small bowel due to that damage. So this is the reason why you see such high mortality with pancreatic plugs forming in cystic fibrosis, and with pancreatic cancer, you get the blocking of trypsin in the duct and then inflammation of the pancreas, so acute pancreatitis, which then spreads to surrounding areas of the body, causing widespread damage and shock and eventually death. The other important thing that the pancreas does is secrete bicarbonate, which will be added to the duodenum. Bicarbonate is made in an active transport process between the bloodstream and the cells within inside the duct. essentially making bicarbonate to be squirted along with sodium into the lumen. This is a really important process. It's activated when there is a high amount of acid in the blood, and that high amount of acid is coming from the acid being, oh man, I'm sorry, I had to yawn. They had to be absorbed from the duodenum. So that's going to trigger this process across this barrier and give this large amount of bicarbonate to the lumen to neutralize the presence of acid. Again, the regulating products, here's a nice picture putting it all together. We've got our CCK and secretin. CCK is secreted by duodenal and jejunal cells of the epithelial surface, and you get secretion of pancreatic juice. You also get contraction of the gallbladder, but right now we're talking about the pancreas. Secretin is made by the duodenal and jejunal epithelia in response to low pH, and it causes this nice secretion of bicarbonate. This happens during the intestinal phase, and these will help turn off the gastric phase and then also enhance pancreatic activity. When you give a patient acetylcholine, vagal stimulation causes a minor release of pancreatic juice, which tells you just general parasympathetic function has some regulatory activity as well. Okay, so here we have a summary of everything the pancreas is doing. So acid from the stomach causes secretin release. Amino acids and fats give you CCK. They're absorbed into the bloodstream, and you get pancreatic secretion. You get juices, you get bicarbonate, and you can also stimulate pancreatic activity with cholinergic stimulation. Woo! All right, finally, we've got the liver. So liver from the front, large lobe, small lobe. The canaliculi are on top, these tiny little canals. They are attached to cells that make bile. The bile flows down them, comes down. Sometimes it backs up the cystic duct into the gallbladder. Otherwise, it just flows on down through, the common bile duct, which converges with the pancreatic duct. All right, so bile. Bile is important for a variety of reasons. First, for food reasons, bile helps to emulsify fat, which means it breaks it down into tiny globules. Bile salts attach to large fat globules, and they interfere with the surface tension between them, allowing these smaller pieces to pop off and dissociate. This is key in breaking these down, and then also because you can only absorb into the body for utilization these small little fat globules. So when people have bile insufficiency, you'll see big globs of fat come out with the stool, and if it is pure yellow stool, we call that steatorrhea. Remember, di is associated with water, like diuresis is water production or water loss, like in urination. So diarrhea is water loss through the stool. Steatorrhea is fatty loss through the stool. So you'd see large globules instead of having absorption of fat products. The other thing, besides just emulsification and then absorption of fats into the body, is bilirubin excretion. When we go through the bloodstream, we'll talk about this a little bit more, but when red blood cells break down, they turn into bilirubin. And bilirubin is a yellow product. When it builds up in the body appears yellow, and that is called jaundice. So what needs to happen, A, is that the liver has to be active and making bile to get rid of bilirubin, because bilirubin is excreted through mainly the feces, about 90% through the feces. So you need bile to be able to transport bilirubin physically out of the body. The second thing that the liver needs to do is conjugate the bilirubin. So it conjugates it to glucuronic acid, making it soluble to be added to the feces. It also makes it soluble so it could be added to the excretion in the kidneys and excreted as urine. However, if the liver is damaged, like in cirrhosis or hepatitis, and the cells are not active and you cannot conjugate bilirubin, then it builds up in the bloodstream and it cannot be excreted. The kidneys can't just pick up the slack because you can only excrete conjugated bilirubin. It doesn't matter if it's through the urine or feces. So you need bile to be created so this can be incorporated into the stool. If bilirubin builds up, it's neurotoxic and it can kill you. That's called an endogenous intoxication or an endogenous poisoning. So bile, important. If people have a deficiency of bile salts, the reason these names are on here, there's two different types of bile salts, cholic acid and chenodeoxycholic acid. Those products can be administered to that patient, which is a good reason to know the names of those because you can recommend that. All right, bile flow, again, there are active cells, just called hepatocytes, so cells of the liver making bile. It flows down the canaliculi, and then it comes on down through the duct system. All right, so then we have different messages stimulating these things. We know that the vagus can cause a lot of things to happen. The vagus causes weak contraction of the gallbladder. Not everybody has a gallbladder, but again, if people do, you can have about 50 mils of bile in there, which is quite a lot of bile. So it does a lot of storage. Bile acids from the blood stimulate the parenchymal cells to secrete products to drive bile production. Secretin from the bloodstream stimulates secretion as well. So again, CCK causes contraction of the gallbladder, helps relax the sphincter of Oddi so that when the gallbladder contracts, all that 50 mils is squirted now into the common bile duct so that the sphincter of Oddi can allow the passage of pancreatic juices and bile out through the common bile duct. Hooray! Here is everything put together for CCK and secretin, so you can follow it in pictures, so where it's made, where it flows, and what the activity is. So enjoy. Woo, now we're at the very end. So small intestine secretions. We see enterocytes, so intestine cells, and we see goblet cells. So we already know we're going to have mucus there. So some different things that you'll find in the small intestines that are specialized areas. There are compound mucus cells. We said compound mucus is located at junctures between different things that are connecting. So you'll find compound mucus cells in areas where there's close contact with the stomach and also with the ducts coming from the pancreas and liver. Between the pylorus and the ampulla of Vater is where we find most of these. And this alkaline mucus is made when there's something touching, like the duodenum, meaning the presence of food. And it's also stimulated when something is irritated in the GI tract so that it aids in the movement of that item out of the body. It's also encouraged when the vagus is active and squirting acetylcholine on it. Secretin can cause alkaline mucus production, which makes sense because secretin is driving the intestinal phase. And the main job of this system is just protection. So here's our pyloric glands. Here's the duodenum. This is where you're going to find these compound mucus cells. There is a name for these special cells. They're called Brunner's glands, again, named after the person who found them. The sympathetic nervous system inhibits Brunner's, and that leads to another supposition that high stress leads to duodenal ulcers, as you can get inhibition of these Brunner's glands helping to make this compounded mucus. And if there's a very high P or very high acidic load that's emptied into the duodenum, then there is a likelihood for damage. A duodenal ulcer is the most common location of an ulcer, and they can be really, really big. Okay, other fun things in the small intestine. The crypts of Lieberkühn, which is fun to say. Here's some goblet cells, here's some epithelial cells, and here are some Paneth cells, and they are all in the Lieberkühn crypts. The crypts of Lieberkühn are between the villi in the small intestine, so they lie between each one. They have a secretory property of making mucus. The enterocytes in that area, or labeled here like Paneth cells, they secrete water and electrolytes. It's almost pure extracellular fluid, meaning water that's just kind of around is incorporated into these cells and then spit out into these crypts. The pH here is very high, 7.5 to 8. And secretions are rapidly absorbed by villi, and it helps in the absorption of products. So again, think about the function. The small intestine's job is to absorb products. So there needs to be a breakdown, not digestion, but there needs to be kind of a thinning out of a viscous chyme to help with absorption. Okay, there's also some digestive enzymes that are located in the intestinal walls. They're very, very specialized. They are expressed only when that particular food breakdown product is in contact with the cells in this area. So it's tactile, meaning touch of the food, and it's also signaled by irritation. So anything that would make you sick or uncomfortable. So there's peptidases there that again split up large polypeptides into individual amino acids for deaminization. Sucrases here, maltase, isomaltase, lactase, and intestinal lipase. The rule with these, if somebody is lacking one of these enzymes, that's what people call like an intolerance. So if you are not digesting your foods because you don't have these enzymes, that means that bacteria will digest it. And bacteria make a lot of gas, meaning there'll be more intestinal distension, so a faster gastric transit time. So there's more diarrhea, there's more gassiness, and there's more discomfort. The life cycle of these cells in the area: continual mitosis, basement membrane, crypt to villi, and they live for about 5 days, which is a pretty typical amount of time for epithelial cells and enzymes. All right, we're at the very end, the large intestine. The large intestine's function is to prepare feces for expulsion. So it needs to get things from point A to point B and also aid in the final absorption of water. In the large intestine, there's many crypts but no villi. You only see those finger-like villi in the small intestine. There's also no enzymes being created in the colon, only mucus. And that mucus is created mainly by tactile stimulation. There's something in the colon, so the colon makes mucus. If you stimulate the pelvic nerve, so S2, 3, 4, going to the colon, you can also stimulate increased secretion. And this is mainly a protective mucus so that fecal adherence can occur, and also transit occurs in a nice way. And that's the very end. Oh my goodness, we're done. All right, so sample board question. The parietal cells of the stomach secrete mucus, water, pepsin, or hydrochloric acid. I really hope you know the answer to this by now. If you don't, please come see me and we'll talk about it. Okay, see you all later.